The last topic we have to cover in the brain is brain protection. The brain is protected by the bones of the skull, bone membranes called meninges, a watery cushion called cerebrospinal fluid, and the blood-brain barrier. So the brain is protected by the bones of the skull, brain membranes called meninges, a watery cushion called cerebrospinal fluid, and the blood-brain barrier. So first we're going to focus on those brain membranes called meninges. The meninges, or the brain membranes, are going to be three connective tissue coverings that lie just external to our central nervous system organs, so the brain and the spinal cord. So our meninges are three connective tissue membranes or coverings that lie just external to the central nervous system organs, the brain and the spinal cord. The meninges function to cover and protect the central nervous system. They also protect blood vessels that are serving blood to and from the brain. They enclose venous sinuses, which are collection points for blood leaving the brain and heading back towards the heart. And they contain cerebrospinal fluid. So the meninges of the brain function to cover and protect the central nervous system. They protect blood vessels serving the brain. They enclose venous sinuses, which are these collection sites to return blood from the brain to the heart. And they contain that watery cushion called cerebrospinal fluid. From external to internal, the meninges are the most external dura mater. So we can see our dura mater right here. The middle layer is the arachnoid mater, shown here. And then the innermost layer is the pia mater, shown right here. So from external to internal, the brain meninges are the dura mater, arachnoid mater, and the pia mater. In between the arachnoid mater and the pia mater, so arachnoid mater and pia mater, we have what's called subarachnoid space. So the subarachnoid space is the space in between the arachnoid mater and the pia mater. So the dura mater is going to be the most external meninge. It functions mainly for protection, so it's our strongest meninge, and it has almost a leathery texture. So the dura mater is the most external meninge or external membrane. It mainly functions for protection, and it has this almost leathery texture. It's composed of two layers. It's composed of an outer periosteal layer. That outer periosteal layer is going to fuse directly with the bones of the skull. So the outer periosteal layer of the dura mater has some similarities to bone. So it connects directly to the bones of the skull. The inner layer of the dura mater is the meningeal layer. The meningeal layer looks more like a true kind of fibrous connective tissue. This meningeal layer is going to be the true external covering of the brain, and it's actually going to extend down into the spinal cord, where it's going to become the spinal dura mater. So the deeper meningeal layer is more of a true connective tissue or a true fibrous connective tissue, so it's going to have less kind of bone-like characteristics. And it's going to cover the external surface of the brain, and then it's going to be continuous with the spinal dura mater of the spinal cord. So the important part about the dura mater is that those two layers of the dura mater, the periosteal layer and the meningeal layer, have a couple of regions where they're not fused. So normally that periosteal layer and the meningeal layers are fused to one another, so that's being shown here. But in a couple of regions, you'll notice that there's some separation between those two layers. So we can see that separation shown right here. So this is our periosteal layer, and we have this meningeal layer right here. So in certain regions of the brain, the layers of the dura mater are not fused and they separate, and they're going to form what's called a dural venous sinus. So a dural venous sinus is going to be a collection point for venous blood from the brain, and it's going to drain blood from the brain and deliver it back towards the heart. So the two layers of the dura mater are not fused in some regions of the brain where they form dural venous sinuses. A dural venous sinus is a collection point for venous blood. So after blood has been delivered to the brain via arteries, it's dropped off nutrients, picked up metabolic waste, that blood needs to be returned to the heart or recycled back to the heart. So the blood is going to drain from the brain, and it's going to drain through these dural venous sinuses. So the dural venous sinuses are going to deliver venous blood from the brain back to the heart. The middle layer of the meninges is called the arachnoid mater. The arachnoid mater here is going to be the more superficial layer of that subarachnoid space. And the arachnoid mater is going to have these almost knob-like protrusions shown here that are called arachnoid villi or arachnoid granulations. So the arachnoid mater is going to have these knob-like protrusions called arachnoid villi or arachnoid granulations. The arachnoid villi or the arachnoid granulations are going to protrude into those dural venous sinuses. The arachnoid granulations or the arachnoid villi are actually going to allow for cerebrospinal fluid that's in this subarachnoid space to be absorbed into the dural venous sinus. So the subarachnoid space contains cerebrospinal fluid, and that cerebrospinal fluid is actually going to be absorbed via arachnoid granulations or arachnoid villi into these dural venous sinuses. The subarachnoid space is then going to contain not only blood vessels that are delivering nutrients to the brain, but it's also then going to contain that watery cushion called cerebrospinal fluid. The subarachnoid space is then going to be flanked more internally by the pia mater. So the most internal layer, or meninge, is called the pia mater. And the pia mater is just going to be this delicate connective tissue covering that is going to be clinging very tightly to the convolutions of the brain. So you can see in this diagram that the pia mater is clinging very tightly to each of these convolutions or infoldings of the external brain. The pia mater is also going to be the site of some really small blood vessels or capillaries that are providing nutrients to our brain structures. So the meninges of the brain protect the brain. We've got the most external dura mater, the middle arachnoid mater, and then the innermost pia mater. The arachnoid mater is separated from the pia mater by subarachnoid space. That subarachnoid space is going to contain cerebrospinal fluid, which is that watery cushion. And that cerebrospinal fluid is actually going to be absorbed into the dural venous sinuses via arachnoid villi or arachnoid granulations, so that the cerebrospinal fluid can be recycled into the bloodstream, and it can then be drained from the brain through those dural venous sinuses back towards the heart. So the cerebrospinal fluid that exists in that subarachnoid space is actually produced in the ventricles. So if you remember when we first started talking about the brain, we talked about not only adult brain structures, but also brain canals. Those canals are these fluid-filled cavities in the brain called ventricles. So ventricles are fluid-filled cavities in the brain. These fluid-filled cavities in the brain contain cerebrospinal fluid. So the ventricles of the brain include the two lateral ventricles. These are those two C-shaped structures. So we've got two lateral ventricles. The two lateral ventricles are going to be connected to the third ventricle via the interventricular foramen. So the two C-shaped lateral ventricles are going to be connected to the third ventricle via the interventricular foramen. That third ventricle is going to be connected to the fourth ventricle via the cerebral aqueduct. The fourth ventricle is then going to be continuous with the central canal running through the spinal cord. So the ventricles include the two lateral ventricles, which are connected to the third ventricle via the interventricular foramen. The third ventricle is going to be connected to the fourth ventricle via the cerebral aqueduct. And then the fourth ventricle is going to be continuous with the central canal running through the spinal cord. So the ventricles contain cerebrospinal fluid. That cerebrospinal fluid is going to need to enter the subarachnoid space, and it does so via these apertures or openings in the fourth ventricle. So cerebrospinal fluid in the ventricles is going to enter the subarachnoid space via lateral apertures. So we have two lateral apertures. These are openings to the arachnoid space, the subarachnoid space, excuse me. And then one median aperture. So we have three holes in the fourth ventricle, which allow for cerebrospinal fluid to enter that subarachnoid space. The cerebrospinal fluid is going to be synthesized within each ventricle by a structure called the choroid plexus. So cerebrospinal fluid is going to be synthesized by a choroid plexus, which hangs from the roof of each ventricle. So the choroid plexus is going to be a complex of capillaries in the pia mater and ependymal cells which line the ventricles. So the choroid plexus is going to be a complex of capillaries in the pia mater and the ependymal cells lining the ventricles. Remember that those ependymal cells are a type of central nervous system glia cell or supporting cell. So ependymal cells are a central nervous system glia cell or neuroglia cell, which means this is one of our types of supporting cells. Ependymal cells are going to line all of our ventricles. Ependymal cells are going to have extensive cilia protruding off of that lumen-facing surface or that apical surface. And the cilia are going to function to keep moving the cerebrospinal fluid through the ventricles. So ependymal cells have cilia. When the cilia are beating back and forth, that's going to keep the cerebrospinal fluid moving or circulating through those ventricles. The ependymal cells are also going to be a part of that choroid plexus. So ependymal cells line all of our ventricles. They are also a part of the choroid plexus, which produces cerebrospinal fluid. So the choroid plexus is a complex of capillaries in the pia mater, and then a layer of ependymal cells, which are the cells lining the ventricles. This capillary is very leaky or it's very permeable. So blood serving the brain is going to come through this capillary in a choroid plexus. And because it's so leaky or permeable, the majority of the contents of the bloodstream are actually going to fall through or leak through these endothelial cell junctions, and they're going to be transported across the ependymal cells. So the cerebrospinal fluid is actually just a filtrate of blood. So cerebrospinal fluid is going to be a filtrate of the bloodstream. So we've got a leaky capillary in the choroid plexus. Blood is going to be filtered out of that capillary and across the ependymal cells into the lumen of the ventricles, forming cerebrospinal fluid. So cerebrospinal fluid is just a filtrate of blood. Cerebrospinal fluid is going to mainly contain some smaller molecules, so glucose, oxygen, vitamins, and ions. Once that cerebrospinal fluid is produced by the choroid plexus, it's going to enter the lumen of our ventricles. So the choroid plexus of each ventricle is going to produce cerebrospinal fluid. So once that cerebrospinal fluid is produced and has entered the lumen of each ventricle, the ependymal cells lining our ventricles are going to have cilia that are going to be beating back and forth, keeping that cerebrospinal fluid moving through our ventricular system. So the cerebrospinal fluid is going to flow through the ventricles. It's then going to enter the subarachnoid space via the median and lateral apertures. So cerebrospinal fluid is going to be produced by the choroid plexus of each ventricle. That cerebrospinal fluid is then going to move through all of our ventricles. So it's going to flow through the ventricular system. It's going to enter the subarachnoid space via the median and lateral apertures of our fourth ventricle. These are just holes in the fourth ventricle that are going to lead to that subarachnoid space between the arachnoid mater and the pia mater. The cerebrospinal fluid is then going to flow through the subarachnoid space. The cerebrospinal fluid functions as a cushion to protect the brain, but it also functions to provide nutrients to the brain structures. So the cerebrospinal fluid is going to flow through that subarachnoid space, and it's going to function to be this kind of watery cushion, as well as to provide nutrients to all of our brain structures. So it comes into very close contact with our brain structures, allowing it to provide nourishment. So cerebrospinal fluid is going to flow through the subarachnoid space. We can see that all along here. And then eventually, that cerebrospinal fluid is going to be absorbed into our dural venous sinus via arachnoid villi or arachnoid granulations. So after the cerebrospinal fluid has circulated through that subarachnoid space, so it's moved all around that subarachnoid space, it's then going to be absorbed into the venous bloodstream in the dural venous sinuses. The dural venous sinus is then going to recycle blood from the brain and allow that blood to be drained back towards the heart. So the dural venous sinuses are then going to drain blood from the brain back to the heart. That blood is going to include absorbed cerebrospinal fluid. So at any given moment, we have about 150 milliliters of cerebrospinal fluid in adults. But that 150 milliliters of cerebrospinal fluid is going to be replaced or recycled about every 8 hours or so. So over the course of a day, about 500 milliliters of cerebrospinal fluid is going to be formed by our choroid plexuses. So the cerebrospinal fluid is being continuously formed by the choroid plexus. It's going to enter the ventricular system, enter that subarachnoid space, circulate through the subarachnoid space, providing nourishment to our brain structures, and then be recycled into our venous bloodstream at the dural-venous sinuses in order to be drained from the brain back to the heart. The dural-venous sinus in the brain, or the main dural-venous sinus, is the superior sagittal sinus. So cerebrospinal fluid is going to enter our venous system at a specific dural-venous sinus called the superior sagittal sinus. So this superior sagittal sinus is going to drain blood from the brain, and it's going to be a very obvious structure that you'll also see in lab. We can see it right here. So we've got our superior sagittal sinus. This is very, very large venous sinus running all through the center of the brain. To provide a little bit of clinical relevance, when cerebrospinal fluid is produced faster than it drains, it's going to build up in the ventricles and cause hydrocephalus. So when cerebrospinal fluid is produced faster than it drains, it builds up in the ventricles and it causes hydrocephalus. Hydrocephalus is literally known as water on the brain. So hydrocephalus means water on the brain. So there's this accumulation of cerebrospinal fluid that ultimately increases the pressure being exerted on brain structures. This is extremely dangerous because it can ultimately cause damage to our brain structures. When this happens in children, because the sutures of the skull are not fused yet, it can actually cause the entire skull to expand. That's being shown in this picture here. So in hydrocephalus, we need to relieve that pressure, and to do so, we insert a shunt in order to drain that fluid from the brain, or drain that fluid from those ventricles. Hydrocephalus can be caused by any obstruction to the normal cerebrospinal fluid flow. So that could potentially be caused by blood clots in the ventricles. It could be caused by a tumor, meningitis, or potential trauma. So hydrocephalus is caused by any obstruction of normal cerebrospinal fluid flow. It can be caused by blood clots, trauma, meningitis, or a tumor. The final aspect of brain protection is the blood-brain barrier. The blood-brain barrier is this selective barrier composed of tight junctions between capillary endothelial cells and an underlying layer of supporting cells called astrocytes. So the blood-brain barrier is a selective barrier. It's composed of endothelial cells bound by tight junctions, and then an underlying layer of supporting cells or glia cells called astrocytes. The blood-brain barrier allows for the brain to maintain an extremely stable internal environment. So in other words, it's not going to allow the brain to be exposed to the same chemical variations that the rest of the body is exposed to. So normally the concentrations of amino acids, of hormones, of different proteins and so on are constantly changing in the bloodstream in response to what's going on in our body at that moment. So for example, if you're exercising versus if you just ate, that's going to produce different concentrations of various chemicals in our bloodstream. We don't necessarily want our brain to be constantly responding to those internal variations. We want the brain to have this really stable internal environment. And so the blood-brain barrier is going to only allow for certain substances from the bloodstream to actually reach our brain structures. So the blood-brain barrier acts as a selective barrier, allowing only certain substances from the bloodstream to actually reach the brain. So for example, the blood-brain barrier is going to allow for gases, glucose, and fat-soluble substances to pass through that barrier, so across those endothelial cells and across that layer of supporting cells. Whereas the blood-brain barrier is not going to allow proteins, toxins, blood cells, or blood-borne waste to pass through. So in general, our blood-brain barrier is going to allow smaller substances to pass, but it's going to prevent larger substances. So I've got two diagrams of our blood-brain barrier to kind of illustrate how it acts as a selective barrier. So first we can see we've got a bunch of blood vessels serving the brain. If we were to zoom in on the walls of those blood vessels, you would see that the blood vessels have these capillaries lined with endothelial cells. The endothelial cells are bound by extremely tight junctions. We can also see that in this diagram. So we've got endothelial cells. They're lining that capillary, that blood vessel, and they're bound by extremely tight junctions. So they're preventing things from passing in between those cells. We also have these astrocytes. Astrocytes are a type of central nervous system neuroglia cell, and they're also going to have some cellular protrusions that create this kind of lining along that capillary. So astrocytes have these kind of feet-like projections that are going to line up along the endothelial cells, adding another layer to this barrier. So the tight junctions between the endothelial cells and the feet-like protrusions of our astrocytes are going to allow for only small substances to be transported across those endothelial cells and then into the brain. So small molecules, such as gases, glucose, and fat-soluble substances, are going to be allowed to pass through that blood-brain barrier, whereas larger molecules, such as red blood cells, toxins, proteins, and blood-borne waste, will not be able to be transported across those endothelial cells and enter the brain. Therefore, the blood-brain barrier is allowing for the brain to have a stable internal environment despite the normal chemical variations that exist in our bloodstream throughout the day.